The dialysis and osmosis lab, part A. Part A is all about electrolyte demonstration, which refers to the ability of a compound when dissolved in water to conduct electricity. Conducting electricity, you flip the switch, the light goes on, that's electricity. Of course, that's in a big circuit, it has to make a complete circle. So it comes out of the battery or out of the wall, through the switch, through the light, and then back into the battery, or back into the wall, makes a full circle. And if you can interrupt that circle anywhere, then of course the light is off. If you connect it, the light goes back on. We're going to do the same thing here, but instead of a switch, we're going to have different solutions to see if different solutions can connect the wires together to turn that circuit on. So let me demonstrate this for you here. Here's our apparatus to test different solutions. We simply plug it in, normal plug, 120 volts, put it in there. And now we've got the light bulb, and you can see here we've got two wires coming down here. So the electricity is coming through the wire, and it's coming down into one of these. If we connect this is like our switch. If we connect those two wires, it'll go back up through the bulb, back, and the light bulb will turn on. And I can actually demonstrate that just with a normal screwdriver. If we connect the two wires together, the light goes on. All right? And then if I take it away, it goes back off. And so there's my switch, on and off, like that. Of course, I'm connecting it with a piece of metal. The electrons can flow through the metal, and again, that's the same as connecting those wires. We're going to try it with solutions. Will the solution also have the ability to allow that electricity to flow from one wire to the next? All right? And we'll just go right down the list that's right on your data sheet there in order, trying these different solutions. So the first one, 0.1 molar sodium chloride. Molar means moles per liter. That's a measure of concentration. That's simply referring to how much sodium chloride is actually dissolved in this liquid. So this is again at 0.1, that's the level. I could go higher, I could go lower, if I put more salt in. Sodium chloride is just table salt, right? So, let me get this DI water out of the way. Okay, here we go. A solution of sodium chloride, 0.1 molar. I'm just going to lift this up so the solution touches those two wires, and then we'll see, does it actually make contact. And you can see the light goes on very brightly, not a problem at all. So when I go up and those go into the solution, it connects them. So electricity is allowed to flow through that solution. That's what that means. I would classify that, therefore, as a strong electrolyte. It strongly allows the flow of electricity. All right, the rest I'm not going to classify. I'm going to let you classify it as strong, weak, or non-electrolyte. So a strong electrolyte is like what we just saw. A weak electrolyte, well, just a little bit of electricity would flow, and so the light would go on, but maybe not so bright. A non-electrolyte does not allow the electricity to flow, and so the light would stay off. All right, so I will rinse this off. I just got some DI water here just to make sure that our contacts are clean between each trial. Our next solution is 0.01 molar. So our first one was 0.1, this is 0.01, so there is 10 times less salt, sodium chloride, in this solution. All right, here we go. It goes on, but I can tell, maybe you can tell anyway, it's not quite as bright. Here's the 0.1, just try to show that again. Here's the 0.1. Here's the 0.01. So you can see it is working as an electrolyte, but not as strongly. Third one, we're going to drop it down another tenfold. So now it's 0.001. So 10 times less than the second solution, or 100 times less than the first solution. Again, let me rinse it off just to make sure there's nothing hanging on there. All right, and now 0.01 molar sodium chloride, we put that in. And I don't know if you can see that or not. It is just barely turning that light bulb on. Just barely. So we are conducting electricity, but nothing like the other ones, obviously. The amount of sodium chloride then, 0.1 or tenfold less, or tenfold less again, the amount of sodium chloride is affecting how much electricity that solution will conduct. Right? From a very, very dim to pretty darn bright. All right, So next on our list is 0.1 molar hydrochloric acid. This guy here. What's the difference? We looked before at 0.1 molar sodium chloride. And I would bring that back here just to remind you. The very first solution, 0.1 molar sodium chloride, very bright. Let me rinse that off. And now my 0.1 molar, so the same concentration, but 0.1 molar hydrochloric acid. Very bright, maybe even brighter, I think, perhaps. So the nature of the chemical, what it actually is, will affect the ability to conduct electricity just as much as its concentration. Next, oops, not this one. I'm going to go in order of the sheet. Next is 0.1 molar sodium hydroxide, so that's this one. Hydrochloric acid, that was an acid, this is a base, sodium hydroxide. The same concentration, 0.1 molar. Okay, so again, in comparison, here's the acid, 0.1 molar HCl. Okay, and rinse it off. 0.1 molar sodium hydroxide, NaOH. Just about the same, I'd say. Very bright. So they're both conducting electricity fairly well. Both are strong electrolytes. All right, now we go to acetic acid, 0.1 molar again. So we saw what 0.1 molar hydrochloric acid looked like, pretty bright light. This is also an acid. Acetic acid is the stuff that's in vinegar, acetic acid. Same concentration. Let's see how this one will do. See quite a bit of a difference. Yes, the light bulb's turning on, but not as bright as the hydrochloric acid was. Let's see the difference there. Okay. So again, the nature of what the compound is, even if they're at the same concentration, the nature of it, just its identity, will affect the ability to conduct electricity or not. All right, so we're about halfway through the list. Next is ammonium hydroxide, so ammonia. This is the stuff that's in Windex, cleaner ammonium. 0.1 molar again, so we're keeping the concentration the same each time. We're just varying what the chemical is. Okay, here we go. 0.1 molar ammonium. A little bit. Not super bright, but it is conducting it. So again, you can classify that as either a strong electrolyte, a weak electrolyte, or a non-electrolyte. That's up to you to figure out. Okay, next on our list is 0.1 molar sucrose. And here's sucrose, 0.1 molar, same concentration as before. Sucrose is table sugar. Stuff you put in your coffee, your tea, or you bake cookies with, that kind of thing. So here we go, 0.1 molar sucrose. Put it up there, and nothing at all. Nothing at all. That is a non-electrolyte. Okay. Let's rinse that off. Next is 0.1 molar glucose. So here is 0.1 molar glucose. Glucose is blood sugar. Anybody going into the health field, you better know that one, right? Blood sugar. So what's in your blood, how your body actually distributes those carbohydrates to every cell in your body. So glucose, 0.1 molar glucose, here we go. Nothing. Nothing at all. Okay. Ethanol is next. Ethanol is what's in beer and wine. So it's an alcohol. Go ahead and clean this off again. All right, ethanol. Here we go. Nothing at all. So ethanol is a non-electrolyte. Finally, our last two, deionized water and tap water. We all know, don't take your hair dryer in the bathtub with you, right? You're going to electrocute. But here's tap water. Right from the taps of Abilene, Texas. Tap water. Up it goes. And you can see tap water is definitely conducting that electricity really well. And deionized water, That's what I've been rinsing it with all along here, DI water, deionized water. And you see when I put deionized water in, nothing. Which amazes a lot of people. I mean, here's water, it's pure water. Pure water does not conduct electricity. Water only conducts electricity when you have stuff dissolved in it like tap water has. Tap water is not pure, it's got iron and other minerals and such dissolved in it. And that's what makes it conduct electricity. Okay, we made it through the whole list. Okay. All right, so part B, tests for chloride, glucose, and starch. These tests we need to understand what they look like and how to properly interpret them so that we can get to part C and actually look at the dialysis and osmosis part of this lab. So part B is just learning the tests, learning what does it mean to have a positive test or a positive result or a negative result for those three things that we'll be, again, looking at in part C. So here's the first station in part B, the chloride part, the chloride test. Chloride is one of the ions that are found in living cells, and that's why we're going to be testing for it here. And we can use what's called the silver nitrate test to detect chlorides in solution. And we're going to use for positive control 10% sodium chloride. So quite clearly there are chlorides in this solution. We put sodium chloride in it on purpose. For the negative, we've got deionized water. So there's nothing as far as chloride. It's just pure water in this one. And so there's no chloride. So when we do this test, we'll see what the negative looks like and a positive result looks like. All right, so following the protocol, it says 3 milliliters into a small test tube for each one. So here's my water. This again will be the negative. And I want to get down here right to the 3 mark, and we'll get 3 milliliters using our graduated cylinder. Okay, right there. And pour that into our test tube, labeled DI water. And you know what, I'm going to put this right up front so we can see it easily. And our positive control, sodium chloride, 10%. Again, we'll get 3 milliliters of it. Good. And pour that into the second test tube. Again, properly labeled 10% sodium chloride. Always label your bottles and your test tubes so you know what's in them. Okay. Next, it says add 2 drops of 0.1 molar silver nitrate. When the silver nitrate goes in there, the silver cations react with the chloride anions to produce a precipitate, that is to say a solid that's not soluble. So if I put it in the negative, there we go. I don't know if you were able to zoom in on that. Not much happened. Here we're going to go ahead and put in the positive test tube, the one with the silver chloride, and if you're zooming in right down there, watch what happens when I put the 2 drops in. 1 drop, 2 drops. Quite a bit of a difference. You see, I'll let the camera go ahead and zoom right in on those 2 test tubes. On the left, we have a negative. If there's no chloride, that's the result we get. This is our positive. There is chloride, that's the result we get. So when we are testing some solution in the lab for chloride and we do the silver nitrate test, that's what we're looking for. That's be a no and that would be a yes. All right, so we do these ahead of time so that we know what to look for. We know what a negative and a positive is like. So there we go, that's chloride. Moving right along, here's our second station. This one is for glucose. So again, for blood sugar, how do we test for blood sugar? We use something called Benedict's reagent. And again, we'll do the positive and the negative to see what both of those look like. So, first, prepare a boiling water bath. There we go. Got that going already. We've got some boiling chips in there just to smooth the boiling out a little bit. Water, it's just water in there and it's boiling away on our hot plate. We get our negative and our positive just like before. I'll bring our test tube rack over here. The negative will be DI water, And here I've got some in a wash bottle. So I'll just do it this way. 3 milliliters, roughly. Yeah, a little more, but it'll be okay for this test. Okay, so there's our negative. There's no glucose in there, that's just pure DI water. And then for our positive, we'll go ahead and use 10% glucose. Prepare it ahead of time. Remember, glucose is blood sugar. So you could use this kind of a test to see if you have that present or not. Maybe you're looking for whether you have glucose present in urine or something like that. All right, so here's 3 milliliters, and we'll pour that in our second test tube labeled glucose. Okay, so once again, our negative and our positive. Next up says add 3 milliliters of the Benedict's reagent. Okay, so here's our Benedict's reagent. 3 milliliters into each test tube. Nice blue solution, right? Very pretty, actually. That's one thing I've always liked about chemistry. You get beautiful colors in chemistry. Sometimes you get beautiful smells or not so beautiful smells too. But the colors are always great. All right, so we're going to go ahead and add that to our test tube. This is the DI water, so here's our negative side. And it's just kind of mixed together like that. And now we'll do it with the positive. So 3 more milliliters here. Yeah. Go ahead and add that to this test tube. Okay. Well, right now they look identical, but we're not done with the test. If you read further, it says now, heat both of those test tubes in a boiling water bath for 3 to 5 minutes. So that's why we needed this. We'll just go ahead and we'll take them and put them in here. I want to make sure I've got about the right amount of water in there. I want it to be heated, but I don't want it so much that the water is going to splash up and get into the test tube. I think we're looking pretty good. So there's the negative one, and there's the positive one. Put those two in there. And now we give them 3 to 5 minutes. Okay. All right, it's actually only been about 1 minute, but we already see a huge difference between those test tubes. And let me go ahead and take them out. Here's our negative, with just the DI water, and here's that positive with the actual 10% glucose. And so that's really easy to tell the difference, right? No glucose, or positive yes for glucose. Very easy to tell whether that's present or not. So these chemical qualitative tests are really useful. Kind of fun to do with colors. All right, let's move along. Here's our third station. This one is to test for starch. Starch, like glucose, is also a carbohydrate. The difference is glucose is a simple monosaccharide. It's got one unit individually. And again, that's blood sugar. Well, starch is huge. Starch is a great big polysaccharide, huge molecule. Starch is actually the way that plants store extra glucose for themselves. So store it up just the way that we store extra carbohydrates for ourselves. Of course, we do it in glycogen. Plants do it as starch. All right, so let's go ahead and do this test. For the negative, again, we'll use water and 3 milliliters. So this was my water one. Oops, I went way too much on that. I'm going to try to make them kind of comparable, right? So let's try that again. There we go. 3 water. So here's my water test tube. Pour that in there. Okay, and then starch, we have a 1% starch solution. This will be our positive control. Again, take 3 milliliters. Of course, We like to eat starch as well. We do have the ability to digest it, right? And we've got a lot of starchy things like potatoes are well known for lots of starch. Which makes sense because that's a root, and the roots often store the nutrients, and starch is stored carbohydrates. So, yeah, it makes sense. Okay, so we got 3 in each one now. Next it says add 2 to 3 drops of iodine. So iodine actually reacts with the starch to give us our positive results. So let's see what that is going to look like. 2 to 3 drops. So we'll go ahead and add 3 drops first to the negative. So iodine just in water. 1, 2, 3. Looks like that. I can stir it up a little bit so it turns it, with a white background you can kind of see that. Turns it now a yellow goldish color. 3 drops of the one that has starch. 1, 2, 3. Mix that up. Ooh, very different result there, right? You get a very dark purple, and if I mix it together like that, you can see the whole thing there. Very dark purple. So again, a negative versus a positive control, we can clearly see that it's a great qualitative test for starch. All right, so these 3 tests are now going to be used in part C in dialysis and osmosis. Okay, part C. We finally get to the osmosis and dialysis section. And to start us off, I'll talk about that in just a second, but let me get the first step done first before I discuss the point of this. It says obtain 12 test tubes. We've got our 12 test tubes. But step 2, combine 10 milliliters of, guess what, the same solutions we just did in part B. Sodium chloride 10%, starch 1%, and glucose 10%. So we're going to combine those together, 10 milliliters of each one into a beaker and mix it all together. All right, so we're going to have all of them present all at the same time. So let me just do this one at a time. 10 milliliters of the glucose. And as long as I get approximately 10 milliliters, that will be fine. It doesn't need to be right on. Because again, we're just doing this as a qualitative, so if we can see if it's present or not. So there's my glucose solution. The second one is the starch solution. About 10 milliliters here. A little bit more. Use the stopper. That's pretty good. Put that in. And finally, the salt sodium chloride solution, so we can get our chloride ions in there as well. This one I'm dribbling a little bit. That's okay. Sodium chloride is not dangerous. Got a little bit of it on the countertop. That's okay. Pour that down. All right, so here's my solution, and we've got a little magnetic stir bar in there, so I can put it on here and turn this on, and that starts spinning. That'll just mix it up, make sure it's all mixed very well. All right. So why are we doing this? Before we get to the next step, which is about the dialysis tubing. We're doing this to show about osmosis and dialysis. And dialysis, in general, is the idea of some small molecules being able to cross what we call a semi-permeable membrane, whereas larger molecules can't. It's a simple concept. It's the idea if you have a net, say in the ocean, right? A net, and the little teeny minnows or little teeny fish, they can go right through the net because they're tiny, but the big fish and the sharks, they can't because they are too big. They don't go through it. It's exactly the same idea. Small ones can get through it, big things cannot. Right? So semi-permeable membranes are like the net. Our cell membranes are semi-permeable. It's the same idea. Some things can get through, other things cannot. Of course, it's a little more complicated than just big and small. You also have the idea of whether they're charged or whether they're not charged, polar versus non-polar. You even have facilitated active and passive transport. So it's a little more complicated, but it is semi-permeable. down to it. And so we are going to mimic cell membranes by using a semi-permeable membrane called dialysis tubing. Dialysis tubing is a tube, as you can see here. Looks like just a clear plastic tube. It is hollow, and so it's like a garden hose sort of a thing, right? And it is made of semi-permeable material. Right? We've got the soaking in DI water. That's just to rinse them off, and once they become hydrated, they open up into the tube. This one in particular, if I look at the label, it says MWCO 3500. That means molecular weight cutoff 3,500. So if molecules, on average, are smaller than 3,500, they're probably going to be able to pass through this tubing. If they're larger than 3,500 grams per mole, then they're probably going to be too big to pass through it. So it's approximately the cutoff point of whether someone can get through it or not. All right, so we have dialysis tubing ready to go. We're going to put our solution in there and then see if we can detect the molecules passing through this, just like what you might see in an actual cell. I'm wearing gloves because if I can look at this with a microscope, it's more like a net, like I said, with little holes. And if I'm rough with it, if I use something that is going to scratch it, it might punch holes in it, or ones that I can't even see, but still bigger than I want to have. And so wear gloves to protect the dialysis tubing. Here are our actual clips. They just open right up. I'm going to put this down actually and show you. They just open up, and so to seal off the ends of the tube, you simply put one across the bottom and snap it in place like that. And so now the bottom of the tube is sealed. You can do the same thing with the top of the tube. Of course, first we got to get our stuff into the dialysis tube. So I'm going to just go and put this back in here so it doesn't dry out quite yet, and go over here and get a second beaker and fill it with DI water, about 2/3 full or so. There we go. There's some DI water, so that it'll be ready to receive our dialysis tubing once we get it filled up with our solution. And of course, our solution is over here, the one that we just mixed together, right? With 3 different solutions. This has got chloride in it, it's got glucose in it, it has starch in it. We mixed it all together. So we're going to pour this inside of the tubing. Now, of course, how do you get it in there? Probably the best way, first of all, gently just kind of squeeze out the extra water, real gently. I barely even touched it, just kind of slid along here. And then the top here, I'm going to open this up and put a little funnel down into it. And here we go. There's a funnel, and I can actually get it to go down into the garden hose, essentially, like that. And so I can pour the stuff down into it. All right, and so here's our solution, and very slowly I'll pour that down into the dialysis. You can see it filling up the tubing, hopefully. Okay, looks like that. So now our tube is full of our solution. Of course, we're doing this solution. You could dialyze any solution you want. Body fluids, blood, doesn't matter. You could do it and see what would cross that semi-permeable membrane. All right, our second clip, I'm going to take it and put it near the top and clip it on. So there we go. Now we've got both the top and the bottom sealed. Notice I didn't put it right there at the top of the solution either. Very often with osmosis, extra water will actually go in and enter, and if you don't leave a little extra space up there, this will actually burst open. So now it'll fit better. I want to make sure the whole dialysis tubing is submerged down below the liquid of the level of the water. Okay, so there we go. We've got it dialyzing now, so the net is in the big ocean, if you will, and little things might start to go across. Now, right away, now that we've got it in there, it says to immediately take out from the water out here into test tubes, 3 milliliters into 3 different test tubes. Why? Because we're going to test that outside water for those 3 things to see if they're getting it across the net. And so we're going to do that right away. We're going to take about 3 milliliters out of each one. Need a bunch of, oh, over here, Dr. Shin. I think right there, 1 milliliter graduated cylinders in that box on the table. We forgot to get this over here. Thank you. All right. So now here's our transfer pipettes. All right, so 3 milliliters, roughly. That's about 2 squirts or so. So there's 1, 2, and 3. So this is kind of like, so we can test the water of what we're starting with. Put that right there. And then as it dialyzes, that might change over time. I'm going to take this, put it over here on our stirrer, and get my stir bar out. Let me rinse this off very quickly with the DI water here. And drop that down in here. And just very gently turn it on, just to kind of keep it stirred up. There we go. Okay, so we are dialyzing. Again, our solution is inside the bag. We have DI water on the outside of the bag. We took our initial sample out. Right? Now we're going to wait 15 minutes to see if those will start to cross that dialysis bag. While we're waiting, we can go ahead and do our 3 tests for chloride, for glucose, and for starch on our first 3 test tubes. So let's go do that in this first 15 minutes. You remember what the negative and the positive control looks like for the chloride test. And all we did is dropped 2 to 3 drops of the silver nitrate in there, and we saw it right away. So there we go, the silver nitrate test here. 1, 2, 3, dropped it in, and not much happening at all. It looks very clear. Again, compared to a positive test or a negative, I mean, negative for chloride. So nothing has made it through, and no chlorides have made it through at any rate. Very good. Next station, we test for glucose. And you remember, this is where the negative looks like, this is what the positive looks like. We add 3 milliliters of Benedict's reagent, and then we heat it up for 3 to 5 minutes. So let me get my 3 milliliters here. Okay. We'll use test tube number 2 for this one. Pour that in. And then we heat it up for 3 to 5 minutes. While that's heating up, let's go ahead and do the starch test, and then we'll come back and see about the results on this one. So we're here, the starch test. Remember, that was our negative, and that's what our positive looks like. All we do is add a couple drops of the iodine to it. So we'll do that here in test tube number 3. 1, 2, 3. Mix that up. And clearly negative for starch. All right, come back here. It's been going about a half a minute. So far, it's still very blue. We'll give that the full 3 to 5 minutes and see if we get any glucose occurring. Okay, it's been about 5 minutes, and this has been heating. Let me go ahead and take that out of here. And this is, again, the glucose test. Oops, my label just fell off. That's okay. The glucose test lines up fairly well with our negative control. And so all 3 tests from that water initially are negative. Okay, so it's been 15 minutes. We've been dialyzing for that whole time. Now it's time to test that water on the outside of the bag again to see if anything has crossed over. So I'll go ahead and take a sample of that water into our second set of test tubes, about 3 milliliters each. Here's our first one. Yeah, 3 milliliters is about 2 squirts in this pipette. Just grabbing the water from the outside of that dialysis bag. Okay. And now, remember, this was from test tubes 1, 2, and 3 for the chloride, the glucose, and the starch test. They were all negative. Now we've got 3 more. Let's go test those. Okay. All right, so the first one, we'll do the chloride test using silver nitrate. Put a couple drops there in the first test tube to look for chloride ions. All right, so turn this so you can see the numbers. That's 4, 5, and 6. All right, so here we go. You can watch right down there to see what happens as I drop the silver nitrate in. Here is 1 drop, 2 drops, 3 drops. I put 4 drops in, but that's fine. And take a look at that. This is kind of interesting. It might be kind of hard to see on the camera. Let me put it against the black, perhaps. That might be a little bit easier. All right. We don't get a lot of precipitate, but you can see it's definitely cloudy. Very similar to the positive over here. All right. And so after 15 minutes, we do have evidence that the chlorides are crossing the dialysis bag. Cool. All right, let's try the second test. The one for glucose. Has glucose been crossing it? So we'll do tube 5 for that. Again, we add 3 milliliters of our Benedict's reagent, and then we will heat it up for 3 to 5 minutes. So we'll get my 3 milliliters. There we are. Pour that into test tube number 5. This one out of the way. Okay, and do a quick mix, and we'll go ahead and put that into our boiling water bath here. I'm going to go ahead and add a little bit of water in here so we heat the whole test tube. And so that'll take a little longer for it to heat up. So we'll make sure to give it at least 5 minutes, maybe even a little longer, to see if we get that positive test developed. And we'll give it at least three minutes after it starts to boil again. While we're waiting for that, let's go ahead and do our starch test. And that's with this test tube here. We'll add a couple drops of the iodine. And remember, the negative is just that color, the positive is the dark purple color. All right, so a couple drops of iodine. 1, 2, 3. Looks like that so far. Give it a quick mix. And so looks negative for starch. So far we've got the fact that the chloride is passing, the starch is not passing, and then we need to come back over here and again wait for this to heat up and boil for 3 to 5 minutes. Okay, so it has been heating now for 5 minutes at the temperature of the boiling, and so let's go ahead and take it out of there. And there we go. Our result is, again, remember this over here. This was the negative. This is the positive for glucose. So I think it's quite clear that we are negative. So just as we can recap here, initially it was negative on all 3 after 15 minutes. Positive for the chloride, but negative for glucose and negative for starch. Okay, it's been another 15 minutes. Our dialysis has been going. We'll sample it one more time. This was after 0, this was after 15 minutes. So our next set over here, 7, 8, and 9, after 30 minutes. We'll go ahead and get our 3 milliliters out of the water from the outside in each tube. Okay. And then finally in tubes 10, 11, and 12, what we're going to do is to test our solution. So break this back open again and test that as well. So let me open up this clip. There we go. And we'll just dip right down into here and get 3 milliliters in each one of these last test tubes. This is kind of floppy, not the easiest thing to do. All right, here we go. So set that right out of here. Milliliters. Okay, so there we go, we've got that out, and let's go ahead and test our last test tubes now. All right, and our usual test, first for the chloride test using the silver nitrate. We'll do that in the first test tube of each set. Add a couple of drops. So here we go, we watch down here. I add 1, 2, 3 drops here, and then we'll do the same thing on the first test tube from inside of the bag. 1, 2, 3 drops there. Take a look at this. You can see again that milky thing, so that would be a positive test for the chloride, as it was before as well, as you remember. And in the inside of the tube, this is a good question. Did all of the chloride exit the tube, or was there some still remaining in the tube? Well, here's your chloride test from the inside. Okay. All right. Let's do the next one. Glucose. We go ahead and add our 3 milliliters of Benedict's reagent. Here so you can see it. There's 3. We'll put that in the second test tube here, so test tube number 8. Okay. And then another 3 for the other test tube. Test tube 11. So as usual, we need to heat that up. So test tube 8 goes in there, and test tube 11. And we give that 3 to 5 minutes. So while those are heating, let's go ahead and do our starch test. Starch test, we just put a couple drops in quickly and easily. So from the outside of the bag, 1, 2, 3, just like that. And remember, here's your negative and there's your positive, and so that's a negative. And from the inside of the bag, 1, 2, 3. Looks quite a bit different. And so the inside of the bag was definitely positive for starch. So looks like starch never made it across that. It stayed in the bag the whole time. All right, so lastly, this has already started to change, at least one of them. We'll give it the full 3 to 5 minutes, and then we'll take a look at it. Okay, so it's been heating for 5 minutes. Let's go ahead and take a look. This one is from the inside of the bag, and definitely a positive test for glucose. Remember, over here, that's the negative, that's the positive. And it's been heating for a long time, so it actually went beyond that orange color and turned out to really brown. Here's the one from the outside of the bag after 30 minutes. Looks like that. And what I want you to do is look very carefully at that, actually. Because in fact, I'm going to pick it up and bring it right next to the positive and the negatives here. Hopefully you can see that while, yes, it's still blue, it is not a pure negative like the negative control was. You can see it's got a little bit of color into it, a little bit of darkening, a little bit of brown in there. So while that's not a strong test, so it's not a strong positive test, nevertheless, we are detecting a little bit of glucose. So most of the glucose stayed in the bag, but you know what? Some of it was starting to cross. And I suspect that it was just a slower process, and if we waited for several hours instead of just 30 minutes, and then tested it, I bet you it would be even darker and stronger yet. So it is getting across, it's just a little slower.